Sean Decatur, who's now, I think, the 19th president of Kenyon. Sean was born in the same city as I was, Cleveland, Ohio. Went to a rival school of mine. I was at University School. He went to Hawkins, etc. And you can take the boy out of Ohio, but you can't take Ohio out of the boy. Even though he went to Swarthmore undergraduate, Phi Beta Kappa, went on and got a PhD at Stanford in the basically biophysics area. Gone on, I'm not sure I can remember all the things, but Mount Holyoke, and rose to the ranks at Mount Holyoke, went on to become, I think, dean at Oberlin, and now president at Kenyon. I also should point out to you, this person has contributed so much to our understanding of education, particularly with regard to the STEM issues. And he is a, 2003 teacher scholar. And we welcome you to present to us now Aggregating Proteins and Coupled Vibrations, a case for the integration of biophysical, chemistry, problem solving, and the liberal arts.
Thank you, Dick, for that introduction, and thank you all for being here. I really am honored by the invitation to come and speak at this symposium. When I look back, the Dreyfus Foundation has been incredibly important over the course of my career. My very first grant, about 24 years ago, was a Dreyfus Foundation startup grant, and was honored to be a teacher scholar later in my career, and now I'm delighted that now that I'm at Kenyon, the chemistry department at Kenyon has a Boissevain lectureship, and so it's been really great for us to have. So I'm going to talk today, at least starting out, wearing my college president hat instead of my chemistry hat, and then we'll sort of gradually merge them over the course of the talk, because I wanted to at least begin by pointing out the value of the study of chemistry and really the study of the physical sciences, the natural sciences in the context of small liberal arts colleges. One of the things I've appreciated a great deal about the support from the Dreyfus Foundation and really the work that the foundation does is that it also has programs that are aimed at primarily undergraduate institutions and the small private liberal arts colleges. And in some ways, actually, when I look at the mission statement of the foundation itself, I'm very struck by the notion of embracing the chemical sciences as a means of improving human relations and circumstances throughout the world, which I think not only is a powerful statement of the goals of the foundation, but when I think about our aims specifically at Kenyon, at liberal arts colleges, of what we aim to do for students, it's not only kind of educating students in a set of skills that they'll go on to be able to apply after they graduate, or a set of skills that, whether in a job or in a graduate program, but really aiming at developing thinkers who are integrating the concepts that they're studying in the classroom with a set of challenging problems that are aimed at making the world a better place, at having a direct impact on the lives of others. This is my, again, plug for the value of liberal arts colleges, that if you look at the larger landscape of higher education, liberal arts colleges serve a very small percentage of undergraduate students in the US. So there are about 4,400 roughly institutions which offer undergraduate degrees in the US. About 100 are in the category that you would classify as private liberal arts colleges, so about 2%. But if you look in terms of number of students, there are about 17 million undergraduate students in the US in four-year programs, about 140,000 at liberal arts colleges, so fewer than 1%. So on the one hand, focusing on the roles of these institutions in science education seems a bit misplaced given the numbers of students that we're talking about. But when you put it into context, and I'm drawing here from a paper that Tom Check published a few years ago where he did a study of the impact of liberal arts colleges on the natural sciences just from the years of 1995 to 2000, so surveying those years, where he found that in those years, 17% of the PhDs in the physical and life sciences had undergraduate degrees from liberal arts colleges. 27 of the top 50 institutions in yield of students who completed PhDs were small liberal arts colleges. And if you look at the folks inducted into the National Academy of Sciences during those years, almost 20% had their undergraduate degrees from liberal arts colleges. So on the one hand, 1% of the total students, but a really huge impact factor in terms of the long-term impact on innovative and important work that's going on in the sciences nationally. So, the importance of the liberal arts colleges to the scientific enterprise more broadly is clear, and I'd argue that the importance of teaching and learning science within the context of the rest of the liberal arts is absolutely essential for our institutions to carry out their mission. And I'd say, turning back to that statement, so I'm going to talk about two different approaches to the Dreyfus goal of improving human relations and circumstances throughout the world through chemistry. The first is focusing on the selection of the problem that I'm going to talk about. So how do we understand and approach a problem in the context of improving human circumstances? And then the second is the process of solving that problem itself. So how do we engage students as part of the educational process, so there's a very intentional training of student thinking along the way. And the problem I'm going to talk to you a bit about is understanding cataract formation. And this is work that, ironically enough, began just as I was beginning to transition from a full-time faculty member to an administrator. It's actually one of the, really is the last project that we took on in my research lab. But I wanted to highlight it here because it's actually a project that really from beginning to end really had its origins in some observations of students in my lab at Mount Holyoke, that it sort of was pushed ahead by some students when I moved to Oberlin who kind of got over one of the big barriers, experimental barriers, and worked in some collaborations with colleagues at Madison to kind of bring the project to completion. And I think all along the path of this problem, the ways in which undergraduate students actually get a direct, hands-on and immersive experience in solving problems gets illustrated. So what are cataracts? So this is an illustration of a lens of an eye that has been, that has cataract. There's a fogging of the lens of the eye, which makes it difficult to see through on the other end. It's an accumulation of protein in the lens of the eye, aggregated protein that's misfolded. This is one of the depressing parts of this talk in that we will all get cataracts eventually. There are cataracts that are forming in all of our eyes as we speak. When I tell this to 18-year-olds upon arriving to college, it completely freaks them out. We are all, I'm sure, in this room a bit further along than they are on that process. But it's also the leading cause of blindness worldwide. So this fogging of the lens of the eye, it's something that in most industrialized countries can be corrected by cataract surgery, which is just replacement of the lens, extraction of the lens, and replacement with an artificial lens. It's a fairly straightforward procedure that happens. In most of the developing world, though, there's not access to be able to do that straightforward surgery. Is there a way to understand cataract formation and understand ways in which we can perhaps address it by other means, by non-surgical means? It's a particularly interesting problem to think about because the lens of the eye is a pretty accessible tissue in some ways. And so if you were to think about sort of other types of interventions beyond surgery, it's something that seems to maybe present itself. So the goal is, can we understand how they form, maybe with the long-term development of therapies that could have an impact. This is just an example of what are the before and after effects of cataract. So if you are thinking of a picture of normal vision and what that picture looks like in cataract, which is just the fogging of the vision. Interestingly, Monet is one of the more famous folks to write about experience with cataract and the way in which cataract impacted the way he was seeing the world. He actually also had very early cataract surgery, and so it's kind of an interesting case of someone who was very sensitive to the visual world, who wrote about how his eyesight was changing and sort of wrote about both before and sort of during a therapeutic approach to this. So this is he did 2 paintings of the sort of famous Japanese bridge, and early in his career, and then later in his career after cataracts, and he actually writes about this in his journal in terms of the ways in which his viewing of the world was shifting and changing. So let's take a look at the eye. So the lens of the eye is a clear piece of tissue that is right in the front of the eye. The main purpose of the lens is to, not surprising based on the name, to collect and focus light into the eye that eventually will impinge upon the retina and then send a signal down the optic nerve. Because of that, the whole idea of the lens is to be transparent and to focus light. That means that these are cells which typically are denucleated. So they are like red blood cells. They develop very early on. They lose their nucleus, and in the mature lens, these are cells without a nucleus. They are really just bags of proteins. Densely packed bags of proteins that are so densely packed that they have the properties of really a liquid crystal array that help to focus the light as it comes through. Now, because these are just bags of proteins, and there really are no other organelles or mechanisms for regenerating the proteins inside the cell, that means that the proteins inside the lens of your eye are the same proteins that you had basically in utero. Right? These are proteins that have to last a really long time. And eventually, like all proteins, they begin to break down. As they break down, they begin to aggregate, and that's what leads to cataracts. So I said that cataracts are kind of inevitable. We will all get cataracts at some point in time because eventually the mechanism of clearing out unfolded protein within the lens begins to break down over time. This is just another picture of sort of an idea of what a lens looks like and a schematic of the lens structure. So a major protein of the lens is this protein called gamma-D crystallin. It's one of a large family of proteins called crystallins. The crystallins are actually a pretty diverse family. Some of the crystallins, like gamma-D crystallin, are purely there to kind of help to shape the optical properties of the lens, and so they're basically like the elements of making up the liquid crystalline properties inside the lens. Other crystallins have passive chaperone properties, and they are there to, as crystallins begin to break down, to kind of vacuum up some of those degraded proteins. But gamma-D crystallin is one of the major proteins of the lens nucleus. It's a Greek key protein with 2 domains. It is very stable, and not surprising as a protein that has to last for many, many, many decades. The melting temperature of this is about 75 degrees. So you can actually take gamma-D crystallins, put it on your lab bench, and it'll just stay on your lab bench for a very long time. But eventually, it's these aggregates that begin to accumulate and form the major components of cataract. So to think about this, we need to put it in the larger context of the protein folding problem. I often tell students that protein folding is really only a problem to chemists and biochemists. We generally fold proteins all of the time without thinking about it at any given second. Proteins are synthesized on the ribosome as long chains of amino acids. They then have to fold into a specific 3-dimensional shape, and that folded 3-dimensional shape is what gives them their function. This is a process that happens. The cell has many opportunities to dispose of proteins that somehow do end up being misfolded, but generally the protein folding process is very efficient. However, both in vitro and in vivo, once in a while, proteins can get stuck in the unfolded state, or they can begin to unwind from the folded state, and as a result, they aggregate. A protein aggregate that everyone is familiar with is Jell-O. Jell-O is a product of taking proteins raising them up in temperature. It's why you actually have to boil the gelatin first. You unfold the proteins, and then as you cool it back down, the proteins aggregate. They make things gelatin-like. So there are many diseases that are associated with misfolded aggregated protein. As you can imagine, forming jello inside the body is not a good thing. One of the types of misfolded or aggregated protein is called amyloid, and these are proteinaceous deposits that you can find in a wide range of tissues. You can stain them and visualize them in, for example, in brain tissues of patients with Alzheimer's or Parkinson's disease. If you look at higher resolution, these protein deposits are actually made of fibers. These fibers are formed from polypeptides, which begin to assemble into these larger units. So how do you actually, these are big aggregates of protein. You can see them with various forms of microscopy. How can we actually get more molecular detail about them? The technique which we started using in my lab, and this is about 20 years ago, was to use infrared spectroscopy to not only follow protein folding, but also using infrared spectroscopy to get information about protein aggregation. Basically, we look at what's called the Amide I band of a polypeptide. It's primarily the carbonyl stretch of the backbone of the peptide. It occurs between 1600 and 1700 wave numbers. And when you have a polypeptide, when you have more than one peptide bond that are linked together, those carbonyl stretches will couple. The coupling between them is dependent on the distance and the angle between the carbonyls. So basically, it's dependent on the backbone structure or the secondary structure of the protein. So you can follow changes in the protein secondary structure. You can analyze the protein secondary structure based on the infrared spectrum, based on what the vibrations look like. So for example, you can distinguish between, and it is not very clear here. It'll come up a little bit better later on. But an alpha helix, which is at about 1630, a random coil, which is about 1650, and then a beta sheet, which has two bands at about 1690 and 1620. So amyloid fibers give this distinctive characteristic of a beta sheet with bands at about 1620 and 1690. And they're fairly sharp and easy to pick out of an infrared spectrum. So if you have something, this is an example of a spectrum of a peptide that's derived from the prion protein. As that peptide aggregates to form fibers similar to the prion protein fibers that show up in various encephalopathies, you get these signature features in the infrared spectrum. So can we do this to figure out something about what gamma-D crystallin is doing? So we know that this is a protein that's aggregating, and that it's aggregating as part of the formation of cataracts. We know this is what the native structure is like. Can we use infrared spectroscopy to get some information about what the structure looks like when it is aggregated? To do this, first, you need to have a way to induce the aggregation. So I described that cataract formation is something that inevitably happens inside the living tissue, but it takes 70 to 80 to 90 years. My students actually objected to this when suggesting that this could just be the length of time of their undergraduate program. So we needed a faster way to do that. It turns out you can speed up the aggregation process by crystalline, by destabilizing the protein a little bit, putting it in the presence of acid, destabilizes the protein, and instead of actually taking several decades to unfold and aggregate, you can actually observe the aggregation over the course of an afternoon. So this is a solution before lowering the pH and incubating. This is the solution afterwards. It gets more opaque. The student at the time at Mount Holyoke got very excited and called this cataracts in a tube. It's a way in which we could actually begin to get formation of aggregated protein in vitro. We could take some of this out and take a sample, and this was done with a collaborator, Jonathan King at MIT, and do a transmission electron microscopy image of it, and we can see the fibers that are formed there. So you get a fibrous solution, a fibrous organization of proteins that are formed out of that. We could also follow this, so change the pH, measure the infrared spectra over time, and we can see a decrease in the infrared band associated with the native structure, an increase in this infrared band at 1620 that's associated with amyloid aggregate. We could follow that over time and measure the kinetics of formations of these aggregates by looking at or measure kinetics of the formation of the fibers by looking at the changes in the infrared spectrum. Okay, so we know that the protein is, that it is unfolding, it's aggregating, that at least parts of the protein are involved in amyloid formation. Question is, what parts of the protein? So this is a protein with 2 domains, for example. Is it the whole protein that's involved in amyloid formation? Is it one domain that unfolds? Is it both domains that unfold? Can we do something to distinguish that in some way? Now, we're limited by using general infrared spectroscopy as a tool for this because that's giving us information about the structure of the entire protein because we're looking at the coupling of carbonyl bonds across the entire structure. It's not giving us localized structure to pieces of the sequence. But, and we know some information from just looking at some studies that had been done on the relative stabilities of the 2 domains, that the N-terminal domain was slightly more unstable than the C-terminal domain. Is this relating to the amyloid formation process in any way? And again, you can imagine the one domain forming amyloid, the other staying structured, vice versa. You can imagine one being unfolded and forming amyloid, or vice versa. You can imagine maybe both forming amyloid. So do we have a sense of, or a mechanism to distinguish between these? So ideally, we'd want some way to tell the difference between the N-terminal domain and the C-terminal domain. It is very easy to do this in a slide by painting one yellow and the other blue, but is there a way to do this spectroscopically that would allow us to observe the kinetics in some way? And we can do this with isotope labeling. It's a technique that we had done in my lab quite a bit as we studied small peptides. So you can introduce in site-specific C13 labels. The C13 changes the vibration of the carbonyl, so now you get a localized coupling among the residues that are labeled. They show up in a slightly different part of the spectrum, shifted by the isotope shift of the label. It allows you to observe, by observing the isotope shifted band, the specifically labeled residues. So we'd done this in my lab for years. It's actually the work that I was doing at the time of becoming a Dreyfus teacher scholar with small peptides that formed alpha helices, small peptides that aggregate. We chose to do that because it was pretty easy to do. If you're making synthetic peptides, you can put in C13 labels because you can buy C13 labeled amino acids, put them in the peptide synthesizer. It's a pretty straightforward experiment to do. Now, labor intensive because you have to move the labels from place to place, but there you just actually bring in a lot of undergraduate students to the lab, and you can actually do a scan of like 60 different labeled peptide schemes in a way that's straightforward. Now, it's harder to do in a protein that you can't synthesize on a peptide synthesizer. So how would we do this? And this is where the observation of a student in lab who insisted that we should actually figure out some way to do this. I sent her to the literature, and she suggested that the way to do it was to actually express the 2 domains separately. C13 label one domain, leave the other C12, and then tie them back together again at the end by a chemical semisynthesis process. So we split the 2 genes for the N-terminus and the C-terminus, express them separately using an intine-mediated system. We purify them, cleave and purify the 2 different domains, and splice them back together. So you can imagine you just do one in C13, one in C12, splice it back together. You now have half the protein C13, half the protein C12. You can do the same experiment and distinguish potentially which domains are doing which. So we did that. You express one domain, do it so that it leaves a thioester at the end, at the C-terminal end. You express the other protein, but with a cysteine residue at the N-terminal end. Turns out you can mix these two things together. There's a chemical ligation that happens that actually forms the native protein, but now just with the cysteine mutant at one residue. Okay? So we did this, purified the protein, demonstrated that the protein had the exact same melting point and stability of the native protein, so we were good to go. So now you grow up one in C12 media, one in C13 media, ligate the two together, and you get a protein that's got mixed C12, C13. You can do all possible combinations of this. So we found that we could do this. The resolution in a simple FTIR that we were doing in the lab wasn't adequate enough for us to fully observe the changes in the spectrum over time that we would like to do. So instead, we collaborated with Martin Zanni at University of Wisconsin-Madison, who does two-dimensional IR measurements of proteins. They're analogous to a two-dimensional NMR experiment. There's a pump pulse and a probe pulse, and you vary the time in between them to watch how the vibrational excitation evolves over time, and it gives you then a better resolved version of the information you get from an infrared spectrum. So to get used to looking at these, so these are a native protein that is just a standard C12 protein, and then you take the whole protein and you label the whole protein C13. And not surprisingly, you see a 40 wave number shift in the spectrum. Okay, so this is the native protein. Both of these are at pH 7. This is the C13 native protein, 40 wave number shift in the spectrum. You can do the same thing. So these are uniform C12, C13 labeled whole protein. This is now in the aggregate form. So that's the amyloid band. That's the much sharper shifted band. And now when you C13 label, that whole band shifts by 40 wave numbers. So if you can now begin mixing C13, C12 on different sides, okay? And so we can do C13 N-terminal, C12 C-terminal, and then we can scramble, we can reduce the other end, and then you can mix together equal parts of fully C12 and fully C13 and see what the spectrum looks like. And do that both in the native state and do it in the aggregated state. It's a little bit easier to see in the summary. So the full-length C12 protein at low pH, so the amyloid in C12 is at 1615. The amyloid when it's C13 is 1575. Okay? When you mix 50-50, you get sort of bands that reflect kind of a mix of kind of in between those two. If you include a C13 labeled domain at the N-terminus, the amyloid is at 1617. And when you do a C13 labeled domain at the C-terminus, the amyloid is at 1575. So we can distinguish then that the 1575 that we see here is because of the C-terminus that's engaged in the amyloid formation. We can eliminate possibilities. So of these, we can eliminate all except that the C-terminus, C-terminal domain is the one that is engaged in amyloid with an N-terminal domain that's unfolded, that's not in the native state. Okay, so that's all using acid. Can we do something under conditions where they might be a little bit more close to what you would see in actual disease formation? So something that is a different type of induction of aggregation that's less artificial than lowering the pH. And one thought is looking at the damage that can happen from UV light. So UV light is also known to trigger cataract formation. One of the interesting epidemiological observations over the years have been that welders are more likely to come down with cataract and come down with cataract at a younger age. And it's believed to be the kind of close proximity of a bright UV light source to the eyes that is doing damage, and it's doing damage to the proteins. So one thing that we're able to work out is that if you expose crystalline to UV light, you get damage to the tryptophan residues within the protein. We could map out which tryptophans were being impacted using mass spectrometry and identify the sites of tryptophan damage within the protein that you're getting due to exposure to UV light. Can also take out samples of the protein after various times of exposure to UV light, and you begin to see the formation of fibers. So you see some kind of blob-like aggregates, but in the electron microscope, you begin to see fiber formation over time. And you can begin to also map this out by two-dimensional IR, and two-dimensional IR, again, with the segmental labeling. And what we find is that we see the same patterns in shifting spectra of the C13 labeled C-terminal domain upon exposure to UV light. So the UV-induced formation of aggregation is happening on a similar pathway as the pH-induced formation of aggregation, which suggests that there may be a connection between the biological induction of aggregation via exposure to UV light and the types of mechanisms that we're able to see in vitro. So I just wanted to kind of bring it all back to the experience of undergraduate students in this, because with the exception of the two-dimensional IR work, which was done in collaboration, and actually a collaboration which we sent Oberlin students to Madison to help collect the 2D IR spectra, all of the other work, including the genetic work for the semisynthesis, the 2D IR work, the one-dimensional IR work in lab, the work in analysis of this was all done by undergraduate students, students who began working on projects perhaps in their sophomore year and built up experience over time. And the experience that they had is very much taking ownership over a project to help to determine the shape and direction of the project, as well as the actual results and ownership of the results on the other end. So I'd like to just, as wrapping up, to thank some of the folks who were involved in the work. As I said, this kind of dates back to my time at Mount Holyoke, my time at Oberlin. We had collaborators at both MIT and at Madison, and thanks funding, and especially thank the Dreyfus Foundation. And I am open to questions, but thank you very much.
Your C13 isotope 2D IR data and results are beautiful, but have you tried a technique called differential scanning calorimetry to differentiate between the N and the C lobe folding and unfolding?
No, so we haven't done any DSC on this. But yeah, is there experiment in particular that?
Well, I mean, you could express the two domains differently and then see at what temperatures they unfold, and then do the whole protein and then see which one is unfolding and so on. And you could use various agents to denature the protein, not only acidic, with acid, but urea or other denaturing agents, and see what if you could mimic, some other physiological conditions to see where the proteins unfold and under.
Yes. So actually, and we have done stability studies on the individual domains, and not with DSC, but we can actually just do regular, sort of circular dichroism melting curves of the domains expressed individually. We can also look at the formation of aggregates, and so both domains will aggregate if you express them individually. The stability does decrease, so they're less stable than the full length two-domain protein when you express them individually. But it's interestingly that both domains actually show about the same propensity to aggregate when expressed individually. So it's when expressed together, and what we believe is that the interface between the domains is important to help to kind of raise the stability of both. And then one ends up being, and it's curious that one ends up then being more prone to aggregate when they're linked together. We think that likely that's aggregating first, and then the other sort of is just unfolded along with the structure.
Along the lines of, now that you understand what is causing the unfolding, or at least which side is unfolding first, does this give you some insight into like possible alternative therapeutic methods aside from, you know, wearing a good pair of sunglasses and getting the surgery later on down the road?
Yeah. And I think that what I think this suggests that's of interest is the interface between the domains, and in particular then stabilizing the C-terminal domain within that interface. And so that's the place where, you know, I know that there are other groups that are now studying specifically, can we probe with mutation impacts on that, on the aggregation, and then if that can give some insights into some ways in which that interface could be, you know, could be stabilized and by introduction of some exogenous molecule of some type.
Is it, so UV damage is often not chemically reversible in any meaningful way. Is it known what happens to those tryptophan residues upon exposure to UVB? Were you able to figure out what the nature of the damage is? Is there any hope it's chemically correctable?
And I think that quick answer is no. I think it's, you know, it's ring opening damage that then becomes oxidatively messy elsewhere within the protein. So there's not, I think that from UV damage, there's not much of a hope that this is reversible. I'd say that most of the standard non-high UV exposure induction of cataracts is likely not as chemically damaging as the UV induction. So in some ways, it's a more biologically relevant example. It's still pretty harsh treatment compared to the standard cataract formation that we've got going on.
So thank you for this. You've had a remarkable range of experience, so I have a fairly straightforward question. Given what you have learned, how would you redesign universities in this country?
Yeah. In a minute or two. And maybe, so I'll cheat a little bit and maybe focus specifically on science curricula. And I think that more inquiry-based research work early on is critical, and the challenge is how to scale that up in a meaningful way. I also think it's critical not just for students who are going on to research careers and graduate work in the sciences, you know, that the thought process in developing and working through a research project is incredibly powerful for students, and in some ways, from a national standpoint, might be more important for students who maybe, say, go in a policy or finance or business perspective than it is for students who go on in a research perspective. Because I do think the understanding of what it means to generate data, to analyze data, to do it oneself is really valuable. So I think the main thing is actually restructuring science curricula almost completely and making it, you know, I would move, if I could design everything from scratch, to strongly research-driven, inquiry-based projects and make that. I know there's some places that are doing that, but often just for students interested in careers in STEM. I think it's actually just as critical for students outside of STEM to get that same type of work early on.
I love that answer myself, because I think too many agencies say, oh, it has to be a junior or senior that would put money into the support for doing some type of independent research, and when so much can be gained. Question?
I did have a question, which is very closely related to that one. So the results that you showed are very interesting and really show what could be amazing things that can be done with undergraduate research. One stunning statistic you showed was a percentage of NAS members coming from liberal arts colleges. So do you have any sense of why that is? Is that a self-selection? Is it actually the early impact? And then I guess related to that question, how do you scale that if that's only 1% of the students?
And so I think some of it is the the early immersion and exposure to sort of independent problem solving, right? That I think is more likely to happen at places that are small, where students are going to be given that responsibility at an early stage. But I also think, and I have no data on whatsoever, so this is pure unbridled conjecture, but that having to do the work in the context of the strengthening one's problem solving and creative work in the humanities, arts, and social sciences at the same time, the fact that these are folks who are working looking at the world through multiple disciplines at an early stage of their intellectual development, I think, is critical. It's interesting that, if you do anecdotal deeper dives, you find that a number of those folks are also, who've gone on to be NAS members who went to small liberal arts colleges. Actually, a good number of them didn't major in the sciences, so they majored in, or they majored in English. They did some work in the sciences, and then they sort of decided that that's what they wanted to do, or they kind of majored in the arts and went into that direction. I actually think as I said, with little evidence here, that there's something about learning in the context where you have to do detailed work in other disciplines that's critical.
Actually, I can just follow, just to make a comment on the data, is that a higher percentage go to graduate school, which I think we've all known, but the percentage of NAS members is much higher than the increase in number that went to graduate school from a liberal arts college. Maybe that's, I don't know, random noise or something,
right.
but it is a large difference in numbers. So that's what I thought was very interesting.
And so there's a paper. This is from a paper. Tom Check did a study of this back in the early 2000s, and he's a Grinnell graduate, so was sort of personally invested in making the case, but.
I hate to bring this to an end, but it's soon time for lunch. Let's thank President Decatur again.
Thank you.